welcome everyone today. So this is a joint seminar of the ARC Training Centre in Predictive Breeding and also the IRTG for accelerating crop genetic gain. So I'd like to start by acknowledging the Turrbal and Yuggera people as traditional owners and for their custodianship of the lands on which we meet today. On behalf of those here and online, I pay our respects to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognise their valuable contributions to Australian and global society. So I'm Peter. I'm our host today. So a few housekeeping things. So today's seminar will run from now until 11:30, but hopefully we'll have some questions at the end. Come on in. We've got the room until 12 o'clock, so we've got some refreshments afterwards, so hang around and have a chat to Kai as well. Just a reminder that today's seminar is being recorded. If you're watching online, you can put your questions in the Q&A tab. Okay. So it's my great pleasure today to introduce our good friend and colleague, Kai. So Kai is a professor and head of department for plant breeding in Geisenheim University in Germany. So he leads a team focused on genomics, phenotyping, quantitative genetics, and biotechnology, with a particular emphasis on grapevine. After completing his PhD in quantitative genetics at JLU in Giessen in 2016, he was a senior research fellow here at UQ and also an ARC DECRA research fellow. He did that before returning to Germany to establish a new research program in horticultural breeding. So his current research develops multi-omic strategies to accelerate the improvement of yield, quality and disease resistance in horticultural crops. And of course, many of you will know Kai from his time here at UQ, and he continues to have a strong connection to UQ now also as an honorary professor in Profi. So it's my pleasure to welcome Kai up to the stage for your seminar.
Thanks. Thank you very much, Pete, for this really nice introduction, and also yeah, good morning, welcome everybody from my side. It's a pretty special seminar for me, actually. As Pete said, I have a pretty strong connection, actually a bit of a history also with UQ. You know, my office was just across in that building at QBP, working, you know, in Ben's group for a couple of years until I moved back to Germany. And yeah, I guess I also want to thank the both centers, the ARC Training Center and Predictive Breeding, and the IRTG for accelerating crop genetic gain, for organizing this, for inviting me. I'm very impressed by, you know, how professional you set this all up. So thanks very much. So today I'm going to do something different. Probably haven't done this before, so bear with me. So I was trying to mix up some content, some highlights, so to speak, from some of the research I've been involved in the past. But I'm trying to mix this up with some career development advice, because I was asked to also give this like a bit of a career development angle, and you know, we'll see how that goes. So the first basically part is about things I've been doing mostly here in my time at UQ. The second part will focus on the things I'm doing right now and hope to be doing in the next couple of years. And then the last shortest part, yeah, it's just my very subjective perspective on a few things related to career development. Now, just before I start, just wanted to make sure, you know, I acknowledge the important people that have basically just, you know, helped me massively along the way, especially in my time here, but continue to collaborate with me, even now in my time in Germany. Very importantly, Lee, who's become a really good friend of mine and a very close colleague over the last couple of years, but also Ben, who I've been working with for, well, in his group as a postdoc in the beginning, and now still continue to collaborate on some really exciting projects. Mark Cooper, which I'm really glad, Mark, you're also here, who's been mentoring me a lot since the last, it's almost like 7-8 years or so. And a whole bunch of other people. Ian let me and my wife sleep in his place, in his house when we moved to Brisbane, which was really great for the first 2 weeks or so. And a bunch of other people. So thanks very much for that. And then I'll try to acknowledge people for the different types of projects that I'll be talking about on the way. So if we think about breeding, and in my role now, I get to talk to non-specialists a bit more often than I used to. So I was used to talking to specialists all the time, but now it's like talking to the general public, talking to people who have no real good understanding of what breeding actually is. So whenever I'm trying to summarize this in one term, in Germany, it's actually just one word. I'd say breeding is just product development, actually. So that's what we're talking about. So very much like developing a new version of a smartphone or, a new industrial product, we develop products in breeding as well. But the products obviously look a bit different. So basically our products are new cultivars. And, we have a very, it's recommended to have a very clear idea of the target product profile. Obviously, the features that the new product has to have. Typically, they are defined by, the industry, agricultural production challenges, needs by consumers, and so forth. But then, the process we go through is pretty analogous. So we design this target product profile, and then we get to create a lot of prototypes, typically through crossing, and then finally we got to market and also maintain these new products, so they ideally make a positive impact in the industry, in the agricultural industry, but then more generally speaking in society as a whole. And the great news is, and this is actually quoting a very smart person who said breeding works. It was actually probably many people said that, but I'm just quoting Mark Cooper here from a workshop or a training workshop that I co-hosted a couple of years ago in Canada. It's like basically the foundation that we're all sort of, based on is the fact that breeding actually works. It sounds very trivial, but it has massive applications. So we as humans have always been able to adapt the crops and the animals we work with to the needs at the time, to the challenges, and the needs at the time. And this is just an example, a data set that I was involved in collecting that just shows breeding progress in wheat over time, in winter wheat, which is one of the most important crops in Northern Europe. And what we see is that we pretty continuously improved wheat cultivars over the last 50, 60 years at rates of about 1% improvement per year. And we've done that under conditions in which we give the wheats a lot of fertilizers and a lot of plant protection, but we've also managed to improve those rates similarly in situations where we give them very little fertilizer or no plant protection. So breeding works, and there's other good examples, but this is just one I've been involved in directly. Now, what are the actual challenges, moving forward, sort of, and trying to push this trend further? There's many challenges, but two of the most important ones probably are the breeding cycles typically are very long. So in my time here, I was working with a lot of spring crops, and breeders would complain about 7-8 year cycle times. Now working with perennial crops, woody crops like grapevine or apple, which take about 40 years, from cross to release a new cultivar. So these breeding cycles, it actually doesn't matter which breeder you talk to, breeding cycles are always too long. And that is one that has big implications in terms of, forecasted gains we can generate, but also at the same time, gains, especially for our staple crops like wheat or corn or other new cereal crops that are actually feeding large portions of the human population, must be doubled in the next short term of like 20-30 years. So these are really critical, or that puts a lot of sort of pressure on the whole field. No pressure, but that just creates a whole new level of relevance for the field of plant breeding and predictive breeding as well. Now, obviously, these centers, or the two centers that are co-hosting this event, they're very much focused on predictive breeding methods. One is called ARC Center for Predictive Breeding. And I'll be talking mostly about genomic prediction in the context of predictive breeding. We can start to mention a few other things towards the end of my talk, but breeding programs have adopted genomic selection worldwide, and it's actually an interesting situation if I compare myself to you, especially the ones that are doing their PhDs at the moment, so I started my PhD in 2013, and at that time, genotyping was pretty expensive still for crops like wheat, for example. I remember my PhD project, we genotyped a panel of wheat lines at around 100 euros per sample. It was like when the 90K array was brand new. So that obviously was super expensive. So probably way too expensive for a commercial program, a small program to adopt, even though, you know, Mark could probably tell you how they already used it in their commercial organizations back then. But at that time, I was interacting very closely, and I've always been interacting closely with commercial breeders. And a lot of them would have just said, it just doesn't work. You cannot predict anything from genomic data. So the concept was still, you know, in many programs, still too new, probably too confusing for some to actually, be adopted broadly. And, you know, there were people I talked to who actually said, big organizations are not making gain because of genomic selection, but despite genomic selection. So they can afford to kind of, you know, play around with that stuff, even though it doesn't really work. You are in a very different situation because as far as I'm concerned, even the smallest programs, even public programs, which are chronically underfunded compared to the big multinationals, they have adopted it. So it's a very different, you know, basis for discussion and also collaboration that you're having now when you talk to people. So predictive breeding is real and it's in full commercial practice. And obviously, most of you would have seen the breeder's equation, the framework which we can use to actually sort of fine-tune our program with regards to maximizing the outcome, the response to selection, which is, you know, kind of at the core of what any breeding operation tries to achieve. Now, the great thing with genomic selection is that it basically tackles all components of this breeder's equation. You can read many books. And you know, we can have a more general discussion also with the more experienced colleagues here as to, you know, how these different components are being tackled. But that's, you know, one of the reasons genomic selection has been so successful and has been implemented so broadly is that it enables you to basically improve all of these components jointly and help you maximize the output that you can generate. Now, if we think about fine-tuning a program within the framework of the breeder's equation, so that it produces as much gain as possible in the short, medium, and long term, because we want to be sustainable also in the way we set this up. Obviously, the first thing that comes to mind is how breeding programs actually set up and designed. And you know, for most cases, I mean, this is basically a generalized structure, but in most cases, we start mixing genetics of parents through crossing. So we generate thousands to tens of thousands of new individuals, offspring from targeted crosses, which we then try to sort out so that after, you know, 5 or 10 or 35 years, like in some of the crops I'm working with, we hope to be able to release a new product, a new cultivar that has a positive impact on the market, in the industry. And as you know, a breeder in that process, and again, you know, we can talk about different sort of nuances of this, but in essence, this is what we kind of iteratively do all the time. And the breeder is confronted with a lot of questions, obviously, but some of the most important questions that a breeder is constantly asking themselves is, how do I perform selection? For example, when do I select, and how do I select the material? And again, this has huge implications with regards to the breeder's equation framework. So the key question, again, I'm trying to break down things to a number of points I can remember. But basically, I mean, and there's other points probably you can think of. But if you think about implementing GS in this context, in this framework, two fundamental questions I think you got to be asking yourself is, what are you using GS for? Do you want to push through that cycle here faster once you've created diversity in order to release products faster? So are you predicting within a cycle, or do you actually try to sort of improve the way you select parents to initiate another iteration of this thing? Because basically what breeders do, they initiate a funnel like this every year. So then you'd be talking about, you know, parent selection and the cross-cycle prediction, and that in the sense or in the context of implementing genomic selection as a technology is a totally different, or can be a totally different thing from you know, the angle of how you approach it. Now, I've brought 3 case studies I'd like to talk about from my time here at UQ, which I think are really cool. The first one's actually so cool it's not even published yet. This is also this is on me actually. And also bad example, you know, for someone who's left their job and didn't manage to write up the paper actually. But yeah, I haven't given up hope, so I might come back for a short sabbatical and write this up. But the results are actually, I think, worthwhile sharing. And this has been done in collaboration with Longreach. Ben was leading the project. You know, had really good discussions and a lot of input also from Mark in this context. So basically, what we are trying to do is test out genomic prediction in a pretty massive data set at the time. So at that time, we had more than 46,000 wheat lines, commercial wheat lines genotyped from the company Longreach. They were tested at 50 locations over 800 trials. And at that time, they were thinking about the way they had set up their multi-environment trial testing system as 3 macro regions. So these are the, you know, colored by red, yellow, and green. And, you know, these were, and again, this is, so for me, coming from Germany, Germany's probably a quarter of Queensland or something in terms of size. So the country is obviously huge. So we're talking about the entire Australian wheat belt almost. And the conditions at which you grow wheat in these regions can vary greatly. And this is just a very simple example, for example, for in-season rainfall and how it differs in those 3 macro regions over a period of almost 8 years, or almost a decade. So it can be quite variable. And that actually also translates into very variable grain yield outcomes of the lines that were tested in those different regions. So for example, you can see that typically macro region 3, so that's the west here, yields the lowest in most cases, but there's some years like 2013, you know, and you might know the reasons for this, where this region all of a sudden, you know, becomes the highest yielding. I don't know what happened in that year exactly, but there seems to be a fair bit of genotype by environment interaction going on. And again, don't need to tell you this, Australia is great or very well known for genotype by environment interaction. But when I was, you know, working with that data set, it probably took me weeks or months to play around with this. But basically, so I learned all the theory around, you know, mixed model analyses of breeding data sets, you know, learned all the theory about G by E modeling, spent a lot of time, for example, also with Mark on this, was trying to use some, you know, some methods like factor analytic mixed models in this data set. The problem really just was I just couldn't get them to work because mainly, I think, because of this. And this is just, you know, flagging or highlighting some of the realities that you're facing when you're moving from an academic data set to a commercial data set. So what is this showing here? So basically, you just see the concurrence of genotypes that were tested across here. So for example, in macro region 1, you see that the lines that were tested in 2012 have basically been thrown out over the course of the breeding program. So they weren't really, or the majority of these lines were never tested again, which makes sense. You know, you start off with thousands in the beginning and you apply very strong selection. So from thousands, you make hundreds, from hundreds, you make dozens and so forth. And that creates this, you know, this really patchy, really unbalanced data that you then try to, you know, use some of your genomic fact analytic models on, and I just, you know, couldn't get them to run basically. But we still thought maybe there's a way to do something about the G by E interaction regardless. So what we've done was we actually had a closer look at genetic correlations that we could, you know, estimate using the genomic data and treating every year macro region combination as a unique environment, you know, and estimating blues for these environments and then estimating genetic correlations across them. And this is what you see here. But we also looked at genomic relationships across the lines as they move through the program. So for like the way you got to think about this is, so for example, lines that were tested in 2012 are fairly related with lines that were tested in 2013 and still with lines in 2014 and so on. But as you move across the scale or across time, you know, the material becomes more and more unrelated, as you'd expect. Again, you know, you initiate a new funnel every year and you throw out a lot of the material every year. So you got this, connectivity issue here. So for example, lines here in macroregion 2 from 2012 are almost completely unrelated to lines, in following years. So we were thinking, can we actually leverage, both our knowledge on genetic correlation structure in this data, but also our knowledge on genomic relationships in this data set to make better predictions than just saying, we're working with a univariate type approach. So we were testing our 2 prediction scenarios. Again, I just explained the top left here, but basically, we were trying to design probably the most realistic and also most difficult prediction scenario. So predicting the performance of new lines in an unseen, untested year. So basically, in scenario 1, we dropped out 2018, both in terms of the phenotype data, but also the genotypes. And then we were trying to predict the performance of those genotypes based on, just 2017 material, just phenotyping and genomic data on those lines, or on 17 plus 18, or 17 to 15, and so forth, down to, using all previous years as a training data set to predict this new environment and the performance of these new genotypes in this new environment. And we did this in a second scenario because we had so many years. We said, okay, can we predict 2019, but following similar principles. Now, this is a bit technical and apologies for that, but I just want to make a short attempt trying to explain to you how we went about the multi-trait prediction approach that we were trying to do. So we were comparing univariate approaches in which we basically derived a single blue across all years, and then try to use that to train the prediction model. That was the simple scenario. We correlated the GBV with the actual macroregion blue estimate. But then we also ran a series of multi-trait models in which we, like I said in the beginning, treated every year macroregion combination as a separate trait. So it's all yield, but we treat them as separate traits. And for me, again, it was amazing that you can actually fit models like this. I didn't think it was possible, but it was great to be based in the Center for Animal Science for doing this, because animal breeders, Ben and all the other colleagues there, they were quite used to working with data sets like that. So basically, what we've done is, so for example, if we want to predict 2019 based on all previous years, you get 7 sets of GBV, basically, and then you can, what are you kind of then using to predict your unseen year? You could just average across those 7 GBV. That was the very basic sort of average approach that we used. But we also used 2 different types of weighted averages. One where we weighted the average based on the genomic relationship of the individual year genotypes with the prediction year genotypes. That was the first one. And the second one was we weighted the average based on genetic correlations. So in essence, saying like in the reference data set, what are the genetic correlations for every year with all the other years? And we were downscaling the years that were kind of extreme, either extremely good or extremely bad, and as shown by a low genetic correlation on average with all the other years. And we were upscaling those years that on average had a higher genetic correlation. So those were the 3 types of multi-trait sort of models that we were applying. And here, again, a busy slide, but it's actually not, hopefully not too difficult to follow. So we were looking at prediction accuracies as the correlation between the actual macroregion blue and the prediction of it. So here you see the range of models. So we tried a univariate base R, a univariate GBLUP, and the 3 different types of multi-trait genomic prediction models. And you can see it. I mean, actually, to be honest, couldn't quite believe that we could predict yield so accurately in some years. Using either model, it actually didn't make a difference. But then obviously it was a lot more challenging, for example, in macroregion 2, in this prediction scenario 2018. And 2018, I think, was also a pretty extreme year. But you could see that in this situation in particular, using a multi-trait approach with different types of weights or a different sort of weighting strategy gave you pretty considerable prediction accuracy still. So overall, some learnings from this sort of first case study were that we can predict yield in Australia, of unseen lines tested in unseen environments with an accuracy of about 0.5 or higher, which is phenomenal. But that's probably also due to the large data set that we've got access to through the collaboration with Longreach. More data can improve the predictions, although it doesn't really do much in most situations. So you can predict, yield as well with just 1 year of training data, as well as you can predict it with 7 years of training data. Again, that's very specific for this prediction scenario context and the macro region we're talking about. But also for us, importantly, it didn't, adding more data didn't decrease your prediction accuracy either. Because, there's a lot of talk in the quantitative breeding community about, fine-tuning the design of training populations, for example. And, I've shown you how over the trajectory of the program, the genomic relationship decreases between prediction lines and training population lines. But, apparently the prediction accuracies weren't really affected by this. So that's also, if you can't think of a really good training population design strategy, you keep adding data, at least in this context, it didn't make the predictions worse. And yeah, multi-trait models were quite interestingly helpful in these really challenging prediction scenarios like this one here, where we basically couldn't predict anything from the univariate models, but we could at least predict something from the multi-trait versions of these models. All right, case study 2, moving from something with an enormous genome, like wheat, like bread wheat, to something with an almost ridiculously complex genome like sugarcane. And that work was really well supported by Sugar Research Australia, and that's been a lot of fun also for me, having never seen sugarcane in real life before in my life. So the idea was to implement genomic selection in SRA's breeding program. So just a few words, and again, I'm no expert in sugarcane. This was the one and maybe only time I've seen sugarcane from very close. Actually, I've seen it again on a vacation trip to Alley Beach. So I've seen sugarcane 2 times in my life. But it's a perennial grass. It accounts for the majority of sugar produced worldwide, and it has a very, very intimidating genome because it's both alloploid, it's autoploid. Modern varieties consist of these 2 species, so Officinarum and Spontaneum. Obviously, both sort of pools bringing in extremely important traits. But the problem is, or the problem of the sugarcane breeding sort of industry is that genetic gain has been extremely low in the last 20, 30 years. And in fact, that's not a problem just for Australia, but worldwide. I could show you some data. Yearly yield performance increases basically plateaued, or were around 0 to 0.3% or so per year, and that is way too low, or was way too low for SRA, and they wanted to ramp this up to say 1 to 2% if possible, using genomic selection. Now, what are the biggest constraints again for genetic gain? We think, well, there's multiple things. Again, sugarcane is tricky to work with, but they have very long breeding cycle times. That takes a long time, also through the perennial nature of the crop. And there's probably large amounts of non-additive genetic effects at play, especially for complex traits such as cane yield. Now, again, we were given this really awesome data set, which I'll show you in a second. And the first idea was, can we actually predict anything, from the genomic data that was generated also with the help of CSIRO at that time? Can we actually predict anything meaningful from genomic data, for traits that are commercially important? Just reminding you, non-additive effects I've already mentioned. It's super highly heterozygous, so very likely, prone to inbreeding depression. And so how, can we? I mean, probably one of the trickiest prediction scenarios you can think of. So that work was led by Seema Yadav, who was a PhD student there at the time. I think very well known to most of you. But she's developed a range or a series of different genomic blood models that were, taking into account different sources of non-additive genetic variants, also some sort of more, non-parametric approaches like RKHS. And very similar to the example before, we're just looking at different traits in the different panels and just looking at how well we can predict those traits in the 4 main breeding regions, or the four main parts of the breeding program that SRA is running. So here, for example, for tons cane per hectare, so that's yield, the yield equivalent in sugarcane, and that is probably the most complex trait to work with. We can see that in the Burdekin region, we can actually predict yield, again, pretty accurately. If we use a training data set of around 2,400 clones, 3 to 4 years of data, and predict, into an unseen year, or unseen clones in an unseen year. But it doesn't really work well in the northern region for some reason. And, that just shows, and also from discussions with the breeders, we knew that, variability in this region is a lot higher, and also like probably the phenotypes were of much lower quality. But we also saw that, if you increase the complexity from a classical G-block model, in terms of adding in, more potential sources of non-additive genetic variants, you actually get a substantial increase in prediction accuracy in some contexts. Again, this is just for the Burdekin region where the phenotype data was probably really good. But you don't see much of a difference for traits like commercial cane sugar, just like the sugar content of the canes or fiber content. And we just hypothesized that this is because these traits are just simpler in terms of their genetic architectures. Now, and this has been a lot of fun, to think about and be challenged with, to be honest, again, the whole concept about additive and non-additive genetic variants. And, I've spent a lot of time also discussing this with Mark and Owen Powell and other people as well. But basically the goal in breeding of any breeding operation is to increase genetic gain. And when we do that in the frame of the breeder's equation, our selection operations target the additive component of the genetic variance. So that is marked in pink up here. The problem or the challenge, though, is that this additive genetic variance can be very highly, it can be very highly affected also by non-additive sources of non-additive sources of genetic effects, as just shown by this figure here. For some reason, the bars are smaller, but it doesn't matter really. So basically, there can be a big discrepancy between physiological genetic effects and the way we pick them up on a statistical level. So we can have a lot of additive genetic effects in theory, and they can translate into a lot of additive genetic variants, but we can also have situations, and there's a lot of really interesting theoretical work that, I'm happy to forward to you, or, Mark has a whole stack of really interesting papers dealing with this, where you have situations in which non-additive genetic effects, or a trait is made up of purely non-additive genetic effects, but you pick most of it up as additive genetic variants. So that's anyway, it's a pretty nice theoretical concept, but with some, I think, or we thought, some implications, because obviously if you target VA purely, the question is like, what are you actually doing to your selection trajectory, and in which direction are you pushing your genetics as you move through your breeding program across time and space. So, and then we thought, okay, we can vaguely, or we can moderately well predict complex traits in sugarcane. That's possible actually, and that could help to improve gain a lot. But then the question also was, how do we best integrate genomic selection in a sugarcane breeding program? Because obviously the way it's been run is more conventional, based off phenotypic selection. And they just wouldn't change the whole thing with us just saying, genomic selection wise. So we were thinking, can we use simulation as a tool to design, or to firstly create a digital twin of the classical SRA phenotypic selection program, and then can we come up with potential strategies as to how to best implement genomic selection, also factoring in things like budgets and time, and also things like or the fact that you can't just simply intercross everything as you wish because of synchronized flowering times and so forth. So we came up with a simulation study. And one important thing with regards to The genetic sort of component that we simulated was that we simulated both an additive scenario in which all the QTL that we simulated were acting additively, but we also simulated a whole range of different scenarios in which we had varying degrees of non-additive gene action. And if you're interested, I think there's a reference to the paper that we published in 2021. There's a lot of details in there. I think it has like 25 figures or something. It has all the details. But to cut a long story short, in the additive model context, we see, in our simulation tells us that we can generate yearly improvements of around 1.3%, which is probably too optimistic because, as we've seen historically in the last 20 years, gain was fluctuating almost around zero. So that, and we know sugarcane is very complex, so the additive model probably wasn't, or was a bit too optimistic. And in this context, two variants of a genomic selection program almost doubled that expected rate of gain. Now, when we used the non-additive model, the situation looked a lot, more interesting actually, but also like a bit more confusing. First of all, we pushed down the, the response to selection per year, or the annual gain, to less than 1%. That was the first thing. But also under both genomic selection strategies, gain was substantially lower, and also obviously a lot more variable across the replicates of the simulations that we ran. So basically, summing up, that under non-additivity, genetic gain is a lot smaller probably. At least if we just apply genomic selection as we do and target this additive component of the genetic variance. This one has to be a bit cautious with, especially if you want to push your selection cycles too fast. If you purely select on the additive component, because you don't really do what that does to the underlying physiological mechanisms. Cool. And then the last case study, and this is who has never heard about FastStack in this room? Okay, you haven't. No, because I figured. I know, and I've been, last time I was here, there was a whole day around, strategies, or a whole seminar day around all the different projects that have come out of this kind of OG project, I would call it. We started in 2017 or 2018 with Ben and again, long reach. So I can quickly just go through this, but basically the situation at the time was, well, if you want to select parents to make a cross as a breeder, so you got 5 wheat lines. So how do you pick the best 2 lines to make a cross if your task is to pick 2 lines to make a cross? Now, all of these lines have a true genotypic value underlying, a true genotypic value that determines the performance of that line in the TPE context that you want to grow it in. The problem is, and we've had this problem ever since, that you can't actually measure that value directly. The best thing we could do in the past is, test and performance testing in samples of that TPE, and by that get an estimate of what the underlying genotypic value is. That is the old school or the conventional method, and it's worked really, really well, as I've shown you in the beginning. But since the last 10 years or so, we've now shifted almost fully to getting even improved estimates of that underlying genotypic value by using things like genomic selection. But pretty much all methods we use, they assume a fully additive genetic model, and they come up with these breeding values by summing up allele effects across the genome, which is a fair enough decision in this context. So you probably pick those 2 lines to make a cross, and their offspring will be good on average. If you just consider their additive breeding values. But we are really interested in asking the question: is there a way to actually include something like this line 4 in the crossing design, which we know has a relatively poor overall breeding value, so you probably chuck it out. But based on genomic information, we know it carries alleles that could nicely complement the others, so that are absent. So that's at the core of the whole idea. So can we develop a framework to combine complementary segments? And that's the FastStack approach. So we need to know, or we need to get an estimate of the effects that those different segments of the genome of your crop of interest has for the different traits that you're interested in. Secondly, we deploy genetic algorithms in this context. I'm sure, and I just saw another paper coming out from Ben's group led by Seema. We're testing out all different other kinds of algorithms to basically do this job of parent selection for you. We've used genetic algorithms most of the times. And then it's really about, coming up with a crossing plan to accumulate or stack up these good alleles in the ultimate stack as quickly as possible. Again, who's never heard of genetic algorithms in this room? I'm serious. So you've probably all heard about this whole story. But basically, if you want to select 50 lines out of 34,000, so 34,000 was the training data set we had access to, the amount of possible sets of 50 parents you can draw from this 34,000 is ridiculous, actually. So that's the number. So basically, it has 240 zeros or 270 zeros or something like that. So it's like a septillion or something. Different ways you can draw 50 parents from this panel. And this is exactly what we use the GA for. So the GA helps us to navigate pretty efficiently in just a few hours through this enormous genetic space to come up with those 50 lines that overall carry as many good segments as possible. So this is when this thing flattens out in the end. So here it's come up with a set of 50 parents that has very good alleles overall, and it can't, improve that subset any further. And this is then the subset we actually work with. And and the great thing about this project, in my mind, or my opinion, is that we didn't just simulate, but we actually also suggested crosses to the commercial partner we were working with. So it's so fundamental to be able to work with long reach for many years. So basically, this is the timeline. So we started nominating crosses in 2018, actually made a double cross between F1 lines, which is kind of pretty outrageous for classical wheat breeding. Like you'd normally never do this, but we've tried it anyway. And then, pushed them through single seed descent, seed bulk up, and did the first round of testing in 2022. I know it's just, 1 year, 4 locations, but here are some of the results. And Eric Dingler-san made this beautiful figure, but I'm just very impressed. So here, the blue bars are the fast stack lines that were yield tested in this year, these 4 different locations. And the red bars are the best commercial checks the company, is always testing. So including their best other conventional lines plus the best commercial checks they can buy from their competitors. And so we see, yield increase of the fast stack lines in that year of between 7 to 50%. Now, this is obviously a bit of a probably too also optimistic view because, we've selected on yield only. So for the wheat industry, quality is probably as important as yield. So we don't really know, I think, at this point, but Ben can maybe share some more insights. But I'm not sure how they perform for quality. That's the one thing. The other thing is we nominated crosses that just look really funny in the field. They grow really tall. They have agronomically speaking zero, like a value of 0. Like you couldn't really produce them. But that's that's just because we trained. The only source of information we've had on this panel was yield and genomic data. So you're kind of making all these funny new lines that breeders just chuck out instantly. But there's apparently some really good genetics or some potential in there, and that was really cool to see and really promising. Okay, I'll try to speed up. So this is this part is nowhere near as long as the first part. So I was focusing a bit more on probably at least half, 2/3 was the first part. So what am I doing now? Just sort of advertising some of the work we do. As far as I'm concerned, no one's really working with perennials here. Well, apart from sugarcane, or are you? Is anyone working with fruits or? Okay, well, we'll make. Oh yeah, Christine. Hey, yeah, no, very good. Because perennials are great to work with, But just a few words on the place I'm now based at. So I'm in Germany. So our uni is very close to Frankfurt, actually, especially for Australian standards. It's a 45-minute drive to the airport. It has really nice towns around. It's like embedded in one of the oldest wine regions in Germany, probably also in the world. So people have been producing wine there for more than 1,000 years. Wiesbaden is a beautiful town. Our uni is quite different to UQ. We only have 550 staff, 45 professors, and somewhere around 1,700 students, which is very small. But also you got to consider that we are very specialized. So pretty much a large portion of the work we do focuses around, or has a focus around, the production and processing of horticultural crops, mainly grapevine, but also fruits, some vegetables and ornamentals. So we are very, very targeted in terms of the things we do there. Now I'm fortunate to head the plant breeding department. So this is just an aerial view. So we're located here. We're surrounded by grapevine, as you know. So we just want to tease you. We have sunshine there occasionally as well in Germany. We think it's a nice spot. So anywhere, if you're anywhere near our area in Frankfurt or so, just make sure you come stop by and visit us. We're a very friendly team of people at the moment. We're about 44. So this is just a list of the researchers and a couple of PhD students that we've got in the team. But obviously we got a large amount of technical staff as well, engineers that support all different kinds of work that we're doing. So the 4 key areas that we kind of really interested in is predictive breeding, or statistical approaches for predictive breeding. We started in this whole exciting field of digital phenotyping pretty much from scratch. This is really great because phenotyping things like grape bunches or apples or stuff like that is really tedious. So we've made some really great progress with a couple of new colleagues there. Genomics, epigenomics, I've been talking about this for a while now. It's super important. And we have a group that's specialized in biotechnological applications as well. So the goal is to be able to, precision breed or edit things like grapevine or apple in the next maybe 2 or 3 years. Great. Now, just briefly talking about grapevine. So the exciting thing about grapevine is, could talk about the challenges a lot, but the exciting thing is that you're actually breeding 2 parts, or you're talking about 2 parts, because commercially grown grapevine is grafted. So the top part that produces the fruits is very different genetically than the lower part. And with regards to the top part, so we either work with traditional cultivars. 97% of the industry works with traditional cultivars. So your Chardonnay or your Shiraz or your Pinot Noir, these are all traditional cultivars, of which we've got hundreds of commercialized clones. And again, the concept of clone is very strange in grapevine, to be honest, compared to other clonally propagated crops. But these clones are almost like sub-accessions or sub-cultivars of that ancient original cultivars, and these traditional cultivars can be up to 2,000 years old or older. So we have a lot of clonal variation within traditional cultivars, but obviously we also do crossbreeding because that's very efficient, because you can move outside the boundaries of these traditional cultivars and mix up new genetics to create improved varieties. Now, again, trying to break complex things down here, but the 2 main challenges we're seeing here is we have very long juvenile phases. So in grapevine, you can measure fruits probably at the earliest after 2 years after cultivating either a seedling or a cutting, 2 years until this thing produces fruits at the earliest, which is a really long time. And then, so things like apple take probably about 5 years to produce their first fruits. So if you think, genomic selection can really help you, accelerate gain in a spring wheat context. Imagine what this could do in a perennial context. If you could predict fruit characteristics in year 1 after making the cross, instead of waiting 5 years, until this thing actually produces a fruit. So there's obviously, and there's other things as well. MET testing is super difficult. I remember saying to Ben and also other colleagues in the beginning, I feel a bit more like an animal breeder now. I'm working with all these extremely heterozygous individuals that you can't easily move around. So they're basically stuck in one spot. And lots of issues there as well. So obviously a strong need for prediction modeling. And some of the work we are looking at thousands of clones, for example, of our favorite cultivar Riesling. So these are just phenotype data and their distributions across years of measurement at one site at our main experimental station. And you can see for yield or sugar content or organic acids, there's a lot of variation. We estimate heritabilities of up to 0.65. So that's how different these clones actually are. And, genetically, they are Riesling in terms of, their microsatellite DNA fingerprints. But we're also interested in generating or studying how variable they are on the genomic level. So we started sequencing clones of that panel, find more than 1 million mutations. And the thing is, the grapevine genome is tiny. So we're talking about 500 million bases per haplotypes, roughly, or maybe 450. I mean, wheat is what, 32 times bigger. But obviously this grapevine has other problems, like it's extremely highly heterozygous. So all the genomics and bioinformatics work was pretty challenging, but we've done some really great work with Peter Crisp as well. It's trying to study, epigenetic variation in this panel, and obviously trying to bring these two sources together. Now, one example here is, I just really like to show this thing that looks like an iron is actually a 3D scanner that produces point clouds, and we can recreate digital images of bunches through these point clouds. It looks like an old iron, but it's actually worth, or it costs like 25,000 euros or something like 40,000+ Australian dollars. But yeah, so we generate these things very quickly. We can do hundreds of them a day, and then pull out different characteristics from these images, and run a sort of conventional statistical genetics modeling, in this case, to actually map variants that underline important traits that give rise to either compact or loose structures. So the industry wants loose structures because you get less problems with mold infections, for example. And we're trying to understand which component traits are actually driving, architectural components of the bunch. Well, we're also interested in looking at G by E interaction, for example, and this work has been led by Hannah Robinson, who I'm sure you also, or many of you actually know very well. So Hannah's run an FA2 model fitting two relationship matrices, a genomic and an epigenomic relationship matrix, and she's made phenomenal progress ever since she's created these figures. But what we find is that basically a lot of the scale G by E, well, G by year, I should rather say, because these plants are stuck in one spot, is assigned inside this modeling framework, is assigned to the genomic fraction, and a lot of the crossover that we see is actually assigned to the G by to the crossover G by E fraction, which is actually pretty cool. So we believe for the first time we can actually show, or we can start asking questions like, how important is genetics versus epigenetics in the context of a perennial that's been propagated for hundreds to thousands of years? And to our knowledge, this hasn't really been investigated much, also due to a lack of suitable technologies for any perennial crop, but happy to have a discussion around that as well. Cool. Now, what are we doing moving forward? So we're really interested in sort of building new knowledge in terms of, creating more data that captures all the different layers of the complex sort of gene to phenotype map. So talking about genomic, epigenomic, we do a lot of work at the moment in phenomic, data acquisition, spectral reflectance, just being one, but other types as well. And, trying to build capacity to actually be able to also just statistically integrate those different types of data. And Hannah is just, I hope she's published that paper now. It's been accepted like 4 months ago, but there was some, I don't know, some issues with the publisher. It might be published already. If not, it'll be published any day now, in which we talk about a lot of this in a bit more detail, but obviously all working towards, breeding better perennial crops faster than we usually, than we used to be able to. Great. Okay, all this stuff, when this has been a bit of time travel for me as well. Like, you try to put in probably a bit too much stuff, but everything, was so exciting. So I tried to do it nevertheless. Now I've got just a couple of thoughts in the end on career development, as I, understand there's a fair few PhD students also in the room, and people, that are kind of, in the same spot I was probably in, 12 years ago or something like that. But you can feel free to just ignore. This is very subjective. Okay, I know my sample size for my own experience is just one, literally one. So this is just what what I thought has helped me. But feel free to just ignore the next couple of minutes if you think that doesn't really apply to you.
Yeah, they're digital.
Exactly. Yeah, but I think well, to kind of put this in the context of the seminar today, careers and lives in general are super complex, obviously, as you all know, and they're super individual and highly non-additive, highly non-linear, hard to predict sometimes, where you end up. So when I was a student in 2013, 2014, coming from a farm that produces a lot of cereals like wheat, produce a lot of pigs. So we're pig farmers. So we still run that farm back home. I actually always thought I want to be an animal breeder until I met the professor for animal breeding at the uni I was studying at, which was not the best experience, but the plant breeding professor was really good. So I ended up, studying plant breeding instead. But also, like at that time, if you had told me that I'll be breeding grapevine 10 years after, I would have thought you're crazy, actually crazy. Like at that time, I probably didn't even know grapevine breeding exists. So very hard to predict sometimes, wherever you end up at. But and I think we have to sort of break this dimensionality down a little bit. And this is my attempt. So running some sort of principal component analysis of that complex system and coming up with 4 principal components that have helped me a lot along my way. So the first one is, and this has taken me some time in my own project, but you really got to own what you're doing. And I think this applies to undergrads, to PhD students, but also as I'm sort of progressing through my own career and getting older and accumulate more gray hair. You really got to own what you're doing. So take responsibility and become the expert. And that's really critical, and learn the skills that actually enable you to gain independence quickly. In the beginning, it's probably more technical, but as you progress through, it becomes more and more like social skills as well. There's a huge amount of opportunities that you can take to improve on that, depending on what you think you need to learn. But you got to realize no one actually cares about your project as much as you do. That's just the reality of it. And so the sooner you kind of act responsibly for your own stuff, the better. The next one is, I think you really have to have a strategy in place and then obviously work hard. But I put work hard second because in my experience, for me, it was the same. So the first year, 1.5, I worked so ridiculously hard, but actually didn't produce any outcome from this. Like it was, just burning energy and resources up, had no clear strategy. It's super critical to know the rules. Like if you're a PhD student, for example, I mean, you've got the grad school, you know exactly what you have to deliver to be able to graduate. Now, we can have a very philosophical discussion around the whole academic system in general, but that's not for today. But I think it's just really important, you have to know the rules of the game. So what do you need to accomplish to succeed? If you're working on a project or a small part of your project, you're generating data, you got to ask yourself, well, what kind of data am I generating? Where do I want to publish this? Ideally, before you even start generating this data. This is, in my experience, has been really helpful, because at the end of the day, you need to publish 3 papers and you want to do some, you want to be creative, but also strategic at the same time, and then work hard. So most cases, and as well for myself, I've done it the other way around in the beginning. It's not very good. There's a lot of uncertainty. Like I said, very hard to predict any outcome. I think the sooner, this has helped me a lot, the sooner I've realized that things just fail all the time. It was quite liberating, to be honest. I mean, I'm still disappointed. I'd say most of the things I'm trying now with getting grants up and so on, or majority of things I'm trying, they actually fail. But it's, that's okay. So you kind of, you just move on and you try again. I think this is just, and it's the same for everybody. In this context, I think imposter is a really big issue for many people, including myself and everybody, I've actually ever worked more closely with. It doesn't really go away ever, to be honest. Like I'm now just feeling more comfortable just knowing I'm definitely I'm the least smartest in the room, and that's okay. So it's very liberating as well. You can ask a lot of stupid questions, and just realizing that everybody's sort of struggling with this, I think also really helps a lot, and and can help you to keep moving forward. And then the last thing is, obviously, it's all about your network. Actually, this is super important, or has been. That's probably been the main thing for me personally. And the ones I know who ended up in really like nice jobs or found peace in what they do, they had a really good network to support them. So find mentors, that do things that interest you the most. Collaborate widely. Again, you have I'd say if you're in your first, second, even third year, you have no idea where you end up. It was impossible coming from a farm in the western part of Germany. Impossible to think that I'd be living in Australia, actually, having a daughter here who's got an Australian passport. It's impossible to predict sometimes. So just stay open. Anything could happen, I guess. And then, to just bring it back to the predictive breeding theme, I think that predictive breeding, just generally speaking, and you're embedded all in this sort of field, it'll continue to rely on fundamentals. So with all these new, technologies and opportunities popping up, I think this is just really important to remember that a lot of the things, a lot of the frameworks we're still using and will continue to use are grounded in very fundamental theory. And this has implications for the second point as well. Obviously, we have more exciting technology popping up all the time. So, and that'll continue to evolve probably at faster paces, even than in the past. But that makes point one even more important. So understanding fundamentals will help you navigate through that, new technology sort of wave that's probably going to get even bigger. And then inter and transdisciplinarity will probably become even more important in my experience. And I still remember the first time sitting down with the crop physiology team here at QAFI when I was new. It was almost like talking to people in another language, like that you have, like Chinese for me, for example, like I can't speak any Chinese. So I had no idea what they were actually talking about. And I felt like they had no idea what I was talking about either. So really, the sooner you can get exposed to people working in other disciplines, the better, because eventually you'll be ending up in any sort of form in an organization or a company or a group in which you got to interface and communicate effectively with other areas. So the sooner you try to work on that, the better. Yep. And that's probably it. Most of all, you should really enjoy the process. I think it's been a lot of fun. For me, everybody I've worked with, I think you are in an amazing environment, like with these two PhD centers. It's actually quite incredible. So you can, really make the most of this. Just make sure you capitalize on all the connections you can make, all the opportunities you'll get. This environment is amazing, and it's a very rewarding, very exciting, fast, paced, evolving field. So, I just hope you enjoy the process, and I hope to, well, the ones who haven't actually visited us in Germany, you'll come over to our side of the world and visit us at some point. Thanks very much. Cool.
All right, thanks very much, Kai. That was it was terrific to see the spectrum of work there, and that was some really great advice for our students. So I think we've got time for some questions from the audience online.
This was not planned, but we've been emasculating grapevine flowers there, and I was wearing my ARC Training Center and Predictive Breeding shirt. That was also just once I realized, I sent a photo to Lee, and he passed it on. So,
all right, Kai, we got a question from Graham Hammer.
Yep.
For the wheat example, great quantitative genetics, but did you think about value for more granularity on E? And just the 3 microregions.
Yes. Thanks, Graham. Very, very good point. Actually, that's definitely the next logical step that needs to be done, because again, I mean, and as you know better than everyone, we're talking about huge geographies. So spatial variability and also variability across times, across seasons is huge. I think with now improved access, as far as I'm concerned, in terms of modeling frameworks for wheat, for example, in terms of capturing crop growth on the one hand, but also better platforms for capturing data, like weather data and information on the soils and stuff like that. It'd be amazing to revisit that data set and try to improve like you said, try to help improving disentangling the E component of this complex G by E system a lot more than we've done. So we haven't actually really touched it.
Yeah. Question, Ben.
Just a question on that. We've discussed it in the past, but Australia's weather is really, part of it is driven by La Nina and El Nino. Do any breeders consider breeding for those different El Nino and La Nina years? So they've got varieties that perform well in La Nina years, varieties that perform well in El Nino.
That's a good point. And last night I saw a preprint published by Mark, actually. I think, and I just had a quick scroll through. looks very complicated, but I think you're actually touching on this point in that paper. So I'll pass this question on to Mark.
So the answer is yes, it's fundamental. If you don't take into account the La Nina and how that influences the frequencies of the different environment types, you'll just be chasing your tail all the time. So right back in the early 1990s, the whole framework that Graham's getting at, we put that in place to support the deconvolution of the sort of patterns of environmental shift and how that moves around on geography. So a lot of the G by E interaction patterns are actually more interpretable when you use that information. And there is about a 10-year cycle. And you think about how long a breeding cycle is. If you're not thinking about this, you end up developing products with 10 years of data that's not well correlated to the future years. So you've got to actually think about the frequencies of environment types and how you're actually delivering that to farmers. And farmers don't work with just one variety anyway. They're actually working with a package using that understanding. And the other thing is on the environmental shifts, one of the things with Graham's work, with Roger Stone, they actually deliver to farmers predictions of the likelihood of La Nina, El Nino events. And this was at planting time, so farmers could actually decide how they wanted to manage their risk. And sort of understanding the risk-reward sort of decisions by farmers is another component of that. But it's a good question, Ben, and definitely it's fundamental to thinking about the G by E by M state space that breeding programs operate in.
Just one short additional thought on what Mark's just said. That also probably puts a different perspective on managing your germplasm pool. Like you said, if you move through that 10-year cycle very quickly, and as you've seen in the genomic relationship trends, for example, in this 8-year time period, for some regions, you end up with material after 10 years that are very poorly related to whatever you've had 10 years ago. So you probably have to think a bit harder around managing your genetics as well and maintaining diversity. Yep.
Further to that, talking about your perennial crop stuff, how do you deal with breeding for what the environment's going to be in 2050? How do you do it in a grapevine?
Well, grapevine is an interesting one because there's no commercial breeding for grapevine, and the main reason I'm saying this is we're very chronically underfunded. So a lot of the work that's been ongoing both in the public and commercial space for cereals, for major cash crops, we're lagging behind like by I don't know, decades sometimes. So the short answer is we don't really take this into account at the moment, at present, but we're trying to take baby steps in the direction of interacting more with modelers. There's some amazing modeling work going on in grapevine as well. Try taking into account where how's our TPE for grapevine actually going to look like in 20-30 years time, for example. So that is the goal. Hang it up like pretty highly for us now, and we're trying to make baby steps in this direction. But at present, no, there's no actual way for us to proactively breed for future environments. Yeah.
Just sorry, a comment. From when I was working in dairy cattle, we developed a GBV, genomic breeding value for heat tolerance. We actually came up with an index, and we looked at different regions around Australia and their climate predictions 20 years in the future. And we came up with a weight that you would put on heat tolerance to maximize performance in 20 years' time. So there's a nice framework for doing it if you have heat tolerance.
Actually, that reminds me, like we do a bit of like a very rough anticipation of, for example, the trends in our region, where we think, the seasons are getting warmer and warmer. That pushes, for example, flowering time and ripening time earlier and earlier. So we've now made crosses, for example, of cultivars that are probably with cultivars that are probably still too late in terms of their physiological, development. But anticipating that it's probably going to be a few degrees warmer, even on average, in 10, 20 years' time, they'll become pretty important. And also drawing on some of the experience we have with previous cultivars that used to be really good in our region that have moved elsewhere, for example, further up north. So phenology is probably a good point. Phenology we can sort of proactively address, but not with fancy modeling, to be honest, at the moment.
I might go back to Christine, and then maybe if we see if there's any brave students want to
Yeah, speaking of students, I think the advice you gave was really excellent. Very good. Thank you. I really fully agree with that, particularly taking control of the project. But all those dot points were brilliant. So I hope everyone was taking notes. My question was a naive one, and thanks for offering up the opportunity to ask naive questions. I love the point you made about, multiple years and how the prediction didn't really improve. I think in one of your environments was a flat line, and a couple they were changing quite a lot. And that's really interesting. But what about population size? Because obviously with some horticultural crops, especially trees, that's another really big issue. Could you just sort of reflect on that?
Yeah. So to be honest, I feel like we probably got to follow the really great examples that animal breeding, for example, given us, like historically, decades ago, or also human genetics, in terms of like forming and building up these shared platforms and data, repositories. I think that's the only way. So in our country, for example, and actually also with pretty big, or in terms of the group sizes, pretty important partners from France, we're trying to do exactly that, teaming up and building those reference data sets that we can use for predictive breeding applications. Jointly, because there's not, in my opinion, and when we are a relatively big group for the public sector, but there's no way we can build big enough data sets just by ourselves and then test them across different sites. So for example, in Grapevine, there's a network now being formed that plans a diversity panel of about 300 or so accessions, but across 15 different countries or so, to do some proper investigations of genotype by environment interaction, but also, predict effects of alleles under different, growing conditions, and also like in different genotype contexts. So I think that's probably the way to go.
Any questions from students, perhaps career advice? Maybe I could kick you off, Kai. When you were doing your PhD, did you know where you were going to end up? And
No.
do you have any advice for the students kind of thinking what's going to be next?
I was just trying to make that point. No, I have no idea. I actually felt really comfortable. Like after finishing my PhD, I was in Rod Snowden's group, and I had a postdoctoral contract, and it was really good. I actually really enjoyed Giessen. It's like a good life, very good, well funded, and well supported. And I just thought that might also, work.
Yeah.
But then I guess, I always kind of had a bit of like an ambitious side too. And I remember talking to my other PhD supervisor, Matthias Frisch, who said, you got to go abroad, actually. If you, just make it very simple, like, and that at that point, that was kind of a bit of a shock, because I hadn't really thought about doing it, actually. Yeah, and then a year later, we moved to the other side of the world, for us at least. Never been to Australia before, so yeah. But that's totally fine. Very, like I said, very highly unpredictable in some cases.
Yeah, absolutely. We got one more question online, and then we could take one more from the room, and then we've got some refreshments. There's a question, is there a concern that genomic selection can potentially reduce genetic variance over the long term? What are your thoughts on this?
Yeah, and I think there's some really good examples and early investigations on this in cattle. For example, they clearly show this, especially in species context, where things like inbreeding play a big role. Definitely is a problem. But then for plant breeding, probably also in the context of what Mark has said, and I can't actually summarize in a few sentences, but just making sure you maintain useful diversity. To be able, like the technical sort of tools to be able to do that, we're still in the process of developing. Like again, with Kira, I'm just thinking of Kira's project with Ben and others. We're just trying to think of ways to maintain useful variation over a period of time, and not run out of it, pretty much. But that's it's a very important point, because you can run super fast, and also you can run against the wall super fast as well.
Yeah,
try to avoid that.
Yeah, absolutely. Any last questions from the room? Yeah.
Thanks for your points. So especially regarding the career. So I was just wondering, like you said, turning the uncertainty into progress. Like when it's uncertain times, like when, from your experience, if you can share some time where you found like it was not turning into progress and maybe you had to reshuffle your cards somehow.
Yeah.
So like, what's the sign for that?
1.5 years, PhD. I probably thought about quitting. Yeah, definitely, most definitely. Just because I wasn't going anywhere actually. And to be honest, it's not like, you know, this is not meant to say you're feeling great all the time. Actually, you know, it's just more like, it's challenging. It'll challenge you on many levels. And it's also, it's challenging everybody on many levels. I think just being aware of that, I think improves the whole situation, like just being mindful of, you know, that this is the thing. In my case, it really was realizing that I cannot fully rely on someone just telling me you got to do this, this, and that, and then that'll, you know, translate into something. Because I've been doing this for the first year, 1.5, doing just, you know, and in hindsight, that person at the time was probably just trying to keep me busy, to be honest, so I don't annoy him. And realizing, well, this will just go on, and you will not graduate because you will not write a single paper, and that'll be bad. You know, it's like either quit or you just say, well, what's actually expected of me? And then it's like, oh, well, we actually generated that data already. It's like, again, weak genotype data at the time. It's also like, that's why I'm most proud of that first paper, because when I started analyzing that data, for example, I had no idea of how to do it properly. So I was reading population genetics papers from humans, and I was looking at all these figures, and I couldn't make sense of them. So I started recreating those human population genetics figures using my wheat data, and sort of taught myself a lot. But then also like realized, well, no one's going to come to me and say, oh, I'm going to teach you everything in population genetics. But I knew there were some people that were really good at it. So, you know, I pestered them at times, like, but with very specific questions. So I think it's a fine balance between taking responsibility and making sure you also convey that to potential, you know, supporters and mentors. If you show up, knock on the door, say, hey, can you just do this for me? And there's no way, you know, people can do this. But if you show up and say, well, I've tried this, this, and that, and this is exactly my problem, and I know you've done this and that, so can you help me with that? I think this has been really like, this has worked really well for me. But yeah, it's I know it's challenging. It's really about ownership, I think, of your, not just of your project, of your whole situation. And realizing no one actually cares about your project. I mean, it sounds really harsh. No, I'm serious. Like, I mean, and I got grants in. I do care, of course, I do care about the projects. But you know, if you are ambitious and you want to turn this into success for yourself by, you know, the means that you think are important for your success, you gotta, you know, take control and ownership of the project and make sure that happens. No one's going to carry you there. Yeah.
All right, I think that's a great thought to end on. We didn't quite get to all the questions, but we better wrap up in the interest of time and having some refreshments. So please join me in thanking Kai again for left you. That was really terrific. A couple of quick announcements, just to highlight that our next ARC Training Centre seminar is on the 15th of October, and we have a fantastic talk from Emma Mace. And also that these seminars are recorded, so if you're interested, go online and you can watch Kai's talk again, or any of the previous seminars. And that brings us to an end. So thanks, everyone. Stay here and join us for some refreshments. Yeah.
Yes.